Thank you, Julie. And thank you to CFRI, the organizers and all the sponsors, everyone who's really worked hard for a long time to put this conference together. It's great to be here with everyone and to be able to talk science about CF. Okay, just some disclosures. I think the one thing that is sort of relevant is I'm going to talk about a compound called linaclotide, which AbbVie owns the rights to, and I have done some consulting for them, but not related to linaclotide. They did not fund the work I'm going to present. They didn't direct any of it, so there should be no conflicts there. I want to start off and acknowledge the people that really have done the work. It's really an amazing opportunity to work with people in science, and I think as we're doing today, really to get around the room and talk about latest developments. So these are all the people that have contributed to the work that I'm going to talk about today. The work I'm going to discuss really has been spearheaded by Dr. Jessica Sarthy, a research scientist in my lab. So although I won't reference her on all the slides, she really has been really the mainstay for the work. And of course, we couldn't do the work with all of our funding, without all of our funding partners, and CFRI plays an important role in that. So thank you to you as well. So I think this audience doesn't need a lot of background about CF, but what I just want to point out is that really over the last 2 decades, we have really seen a significant increase in life expectancy for those with CF. And so what this has created really is that now we have more adults with CF than children. Respiratory failure, unfortunately, still remains a primary cause of mortality, and as a gastroenterologist, I always remember that as much as I preach about the importance of the GI system. Pulmonary disease still is what is by far killing patients. But digestive complications really are important contributors to the symptoms that individuals with CF face, and even contributes to mortality. We've long known that CF mortality is associated with poor nutrition. This section, I apologize that you can't see it. I can't control how it looks on the monitor here. But really, this is sort of actually newer data that it's looked at individuals over time, and the y-axis is survival probability and the x-axis is age. The dark line is those individuals with CF who are undernourished, and the middle line is those who have normal weight, and then the other line are those that are overweight. And so what you can see hopefully very clearly is those individuals who are undernourished continue to have an increased mortality even in these days. There's a nice study done by it's called the Galaxy study. That was done by the CF Foundation, and this probed both children and adults about what types of gastrointestinal symptoms they may have. And while you probably can't read all of these things that are here, it goes through things like bloating, evacuation problems, abdominal pain, and the whole list. And what I want you to appreciate is that up to about 30% of individuals have moderate to severe symptoms when it comes to these GI complaints. And so that's a significant proportion of people. Well, why do so many people have GI issues? Well, I think it's because CFTR is expressed in a whole different host of cells within the GI tract. So here was supposed to be our epithelium, but here we have epithelial cells which express the enterocytes that express CFTR, as well as a bunch of other acid-base transporters. We have enteric neurons that also express CFTR. We also have smooth muscle cells that express CFTR, right? And so all of these have an interplay. And then even within the enterocytes themselves, we know that different cell types express CFTR to differing degrees, right? So we're not just dealing with one cell type, but we're dealing with many cell types that have different functions. And when we look, and all this contributes to typical symptoms that we might see, such as decreased intraluminal pH, which can impair pancreatic enzyme efficacy and micelle formation, which leads to digestion and absorption of nutrients. There can be ineffective mucus expansion and release. When you have decreased intraluminal fluid, you can get compacted mucus and also bulky stool. You can, through the enteric nervous system and the smooth muscle deficits, you can get decreased neural stimulation of contractions in this hyperconstricted smooth muscle. And all of this can lead to gastroparesis, dysmotility, constipation, and incomplete evacuation, right? And so when you talk to people that have these GI symptoms, it's not just one of these things. It's often a multitude of these complex issues. And on top of that, we have microbiome disturbances, right? As if that wasn't enough. So there are multiple studies now that have shown that if you compare the intestinal microbiome of those without CF, it's very diverse and sort of rich. You take individuals with CF and you have very little, abundance of species you see here. And so the model that it really has been put forth is that you have not only these cell-based deficits in ion transport and contraction stimulation, but also inflammation that disrupts the microbiome. You have diet changes that alters the microbiome as well. And all of these contribute to what has been termed a CF enteropathy. And also the state leads to decreased microbial-based nutrients, which can further affect your dysmotility because the microbiome is a rich source for agents that, these neurotransmitters that stimulate motility. So I want to spend a little bit of time on gastrointestinal pH and CF because as you'll see from my talk that our lab, one of the main focuses of our lab is on bicarbonate secretion and acid-base regulation. It's been well established for more than 30 years that the small intestinal pH is lower in CF than healthy individuals. On the top, we have some data using tethered pH catheters. So these are catheters placed right into the proximal small intestine and measuring the pH from healthy individuals and those with CF. And what I just want to bring your attention to is right here is the transition zone from your stomach to your small intestine. And one of the jobs of the small intestine is to neutralize that very acidic stomach acid, okay? And so that happens in individuals. You have very low pH in the stomach, and it quickly rises as you go into the small intestine. In individuals with CF, it does rise, but then you see it falling, rising, falling, falling. And so it remains lower than this line, which is pH 5.5 for quite a while. And why is that line important? Well, pH 5.5 is typically the pH we refer to as where pancreatic enzymes will actually begin to work. Pancreatic enzymes like to work at a pH of 7 to 8, ideally, but we want them above pH 5.5. The data below here was data from Daniel Gelfand, which he used what we call a smart pill, where it's actually instead of a tethered catheter, you swallow this pill. It's wireless, and as it goes through the GI tract, it measures pH and pressure and temperature. And so here in a healthy individual, you see again, it goes from the stomach into the small intestine very rapidly. And in individuals with CF, you see this delayed neutralization, okay? Well, we wondered, well, what about other segments of the gastrointestinal tract? This is just looking at the proximal small intestine and the duodenum. So we went back to the data set that Daniel Gelfand had, and together with Doreen Patel and Stacy Matthews at St. Louis University, we did similar analysis across the entire GI tract this time. And so here we have, let's say, the stomach, the very first part of the small intestine, the middle part of the small intestine, the end of the small intestine, beginning of the colon, middle colon, and end of the colon right before this capsule leaves the body, okay? And in the clear bars, we have the values from healthy individuals. In the red, those that have at least one G551D mutation and something else on the other allele. And then in the blue, those with F508del homozygous. So if you just look at the clear bars for a second, the normal pattern is you should go from the stomach, increase, it sort of comes back down when you go into the colon, and then it sort of increases again. This is a very typical pattern that we should see. But you can see at many of these stages in CF, not so much in the stomach, but in the proximal small intestine, and certainly then later on in the colon, then in individuals with CF, you have greater acidity of the intestine there. So we've already talked about this a little bit today, but you know, in this era of CFTR modulators, I'm trying to think, well, do we really need CFTR modulators? If you sort of pay attention to the popular press, you might think, no, you know, they're wonderful. They're doing so many wonderful things for most of the people. But I think those of us that both see patients, do this research, and you know, talk to people in the community, we know the answer is yes. So I'm just going to highlight a couple reasons that we think about why we need them. One, we know CFTR modulators have variable effects on lung function, right? That's not just depending on which modulator you're talking about, but that is also, even from clinical trial data, using the same modulator, different people have different levels of improvement. Unfortunately, in all of these clinical trials, there were really very limited GI endpoints. We look at, in the ivacaftor trial, you know, the kilograms, and the others in BMI. And so that's all the data we have from clinical trials as to whether these drugs may be able to have an impact on GI health and CF. When we look at the PROMISE study put up by the CF Foundation, they have results from 6 months of elexacaftor, tezacaftor, ivacaftor. And unfortunately, they found that there was no clinically important or significant difference in symptoms in individuals on 6 months of ETI, and there was no significant change in the stool consistency or bowel movements. We don't really know why exactly that is. There's a lot of questions that remain from that, but I think it tells us that we still need to be continuing to think about more therapies, especially in the GI tract. So our lab has been very interested in targeting non-CFTR pathways for CF therapeutics. Sriram gave a nice overview already of some of our data as to why we think this is important, not just for individuals that may not respond to CFTR modulators or may have to abandon them because of side effects, but because there actually are a lot of individuals throughout the world that have mutations that would not qualify for them. And I think one of the important things to remember is that these new therapies really are creating an increase in the health disparities that we see within our communities, right? And we need to be very vigilant about minimizing those disparities. So for the work that I'm going to talk to you about today, really the studies that started to get me thinking about this was work that was done by some of my colleagues at UC San Diego many years ago, over 20 years ago. And what they did was they took duodenal biopsies, so small intestinal biopsies from healthy individuals and individuals with CF, and characterized the ion transport properties in them. And what they found, first off, that wasn't surprising, is if you give a cyclic AMP stimulus in healthy individuals, you see a big increase in bicarbonate secretion, but you don't see much of an increase in CF individuals. But when you use the heat stable enterotoxin of E. coli, which at the time was used primarily to increase intracellular cyclic GMP, you actually see similar responses between healthy individuals and those with CF. So the question that came out of this was, how does STA stimulate normal amounts of bicarbonate secretion in CF, and could this mechanism be leveraged for the development of new therapies? So to tackle this question, we took a couple of different approaches. We started with wild type or CFTR knockout mice. This is a global knockout. And we used two different approaches. We used an in vivo approach where we isolate the proximal small intestine before the common bile duct and collect the secretions from this area and measure bicarbonate via back titration. We also do in vitro, where we take the small intestinal tissue, remove the seromuscular layer, from so we only have the mucosa, and then we put this in Using chambers to measure short circuit current and combine that with automated pH stat to measure bicarbonate secretion at the same time. And so what we found from this early work was that in these mice in vivo, when you give forskolin, you see a really nice increase in duodenal bicarbonate secretion. And when you remove that forskolin, it starts to come back down. But in CFTR knockout animals, you see no response. This was not surprising. We knew that forskolin stimulated CFTR currents, the currents were dependent on CFTR. Oops. But what we were surprised to see was that when you give STA in a wild type mice, again, you see an increase in duodenal bicarbonate secretion. But here in CFTR knockout mice, you also see an increase then of similar magnitude as wild types, which is very exciting to really show us that you don't need CFTR expression or function for this pathway. We repeated these studies in vitro in our Using chambers. And here you see in control that the black bars are baseline, the clear bars are with STA stimulation, that you get increase in bicarbonate secretion in both wild type and CFTR knockout mice. And then in thinking about how this might occur, we removed chloride from the bath to see if this was a chloride dependent phenomenon. And the reason we did this, and I'm going to talk more about this, is that some of the other acid-base transporters, specifically bicarbonate transporters, require chloride to work. And these are what we call chloride bicarbonate exchangers. And so when we do that in our wild type mice, you continue to see an increase in bicarbonate secretion because CFTR does not need chloride there to conduct bicarbonate. However, in our CFTR knockout mice, now STA is unable to stimulate duodenal bicarbonate secretion, meaning it requires chloride. And on this data suggested that in CFTR knockout mice, STA was able to increase duodenal bicarbonate secretion through chloride bicarbonate exchangers. So fast forward a few years down the road, and we got interested in this compound linaclotide, which at the time had been FDA approved for irritable bowel syndrome constipation. And why we got interested in that, and sorry, this has shifted a little bit, is that if you look at the amino acid sequence of STA and you compare that to linaclotide, they're almost identical. And in fact, the whole purpose for generating linaclotide was to make STA variants. Both have 3 disulfide bonds and very similar structures. So we set out on a series of experiments to see if linaclotide also increased bicarbonate secretion and to understand the mechanism whereby it may do so. So we went back to our in vivo model, and here we gave differing doses of linaclotide and found that as you increase the dose at 10 to the minus 7 and 10 to the minus 5 molar, you get an increase in duodenal bicarbonate secretion, a magnitude that was similar to STA. And then we wondered, well, can this process occur independent of CFTR? So we repeated these experiments now in the presence of a selective CFTR inhibitor, CFTR-INH172. And again, sorry, you maybe can't see it here in the room. This dotted line are the experiments that are done without CFTR inhibitor, and the dark circles are those in the presence of CFTR inhibitor. And I'll tell you, they overlap, they look exactly the same. Okay. So then to prove this further to ourselves, we looked at wild type mice and CFTR knockout mice again. And what you see here in vitro is that linaclotide increases duodenal bicarbonate secretion in vitro, and it does so in a way that increases short circuit current. So we call that electrogenic bicarbonate secretion. However, when we looked at the CFTR knockout mice, we see equal magnitudes of bicarbonate secretion, but here no change in the short circuit current. And then we call that electroneutral bicarbonate secretion. So what we're able to see in the CFTR knockout mouse is that we can get bicarbonate secretion, but it happens with no change in the short circuit current. And this is also supportive of the involvement of chloride bicarbonate exchangers, because typically what happens is you are exchanging one anion for another anion. And so it's electrically silent in the Using chamber, or the voltage clamp system. We wanted to see, does this phenomenon also occur in humans? So we took human duodenal biopsies. These are all from healthy individuals with no CF or duodenal disease. And we measured their responses to linaclotide. And you can see that in duodenal biopsies, you also see an increase in linaclotide-stimulated duodenal bicarbonate secretion. In contrast to mice, we see very little change in the short circuit current. So it looks like in humans may involve a little bit more of an electroneutral process than an electrogenic process. And then because at the time we didn't have any CF tissues to use, we did these studies with and without CFTR inhibitor. And again, you can see that when you have CFTR inhibitor present, you continue to get bicarbonate secretion in response to linaclotide, and there's no change in the short circuit current, which not much existed to begin with. So what we've been able to show both in mice and humans is that linaclotide, just like STA, is able to stimulate duodenal bicarbonate secretion in both individuals without CF, but importantly in individuals with CF, with data to suggest that it's utilizing chloride bicarbonate exchangers. And while we were excited about our initial STA studies, we were even more excited about our linaclotide studies because that's already an FDA-approved drug, right? But we wondered, well, how does linaclotide stimulate CFTR-independent bicarbonate secretion? And so we repeated some of the in vivo studies, now using some different inhibitors. So first off, these experiments all have linaclotide exposure. They're all done in the presence of a CFTR inhibitor. So we're only looking at CFTR-independent bicarbonate secretion. And I'm showing data for either in the presence of a selective inhibitor of one type of chloride bicarbonate exchanger, DRA or SLC26A3, and then another inhibitor, which is not so selective, DIDS, but in this instance, how we've used it seems to be maybe more specific for PAT1 or SLC26A6. And what you can hopefully easily appreciate here is that really only the DRA inhibitor abolished the CFTR-independent bicarbonate secretion in response to linaclotide, and that's shown in the time course here with the dotted line, our CFTR inhibitor, and our circles with CFTR inhibitor plus the DRA inhibitor. Okay, so this made us think that really maybe DRA is the chloride bicarbonate exchanger responsible for the CFTR-independent response. So, but we also wanted to see, there are many ways in which you can alter the activities of these channels and transporters. One is you can activate them that are already at the membrane. The other way is you can traffic more of them to the membrane. Okay, so we wanted to look at DRA trafficking, and so we used, we generated duodenal enteroids. But we used a different technique than most people at the time were using. So most of the enteroid or organoid data you'll see is what we call basal out, where the apical side is on the inside, and here's your lumen. So if anyone's seen those swelling assays, the reason they get bigger is because fluid goes into this lumen and they get bigger. But the receptor for linaclotide or STA, guanylyl cyclase C, is on the apical membrane. Okay, so if we were to use this model, we would have to inject those compounds into the lumen. So, but leveraging a technique developed by Manuel Amieva's lab at Stanford, you can remove the extracellular matrix and reverse that polarity. So now you have apical out enteroids. So we have access to the apical surface. And in the staining, which maybe you can see on Zoom better than we can see in the room, is the red is actually the apical membrane. So it's inside here and outside there. And so we use these apical out enteroids, and in this instance, we've differentiated them for 3 days to make them more villus-like. And we can have control conditions where there's no water control, and then linaclotide stimulation. And what you can hopefully appreciate is this green is DRA staining, is that as in these apical enteroids, there is a bunch of DRA expression, but there's actually very little that's at the apical membrane at baseline. However, when we give linaclotide, we see much more that goes to the membrane, and that's quantified here. So it seems like linaclotide is not only activating DRI, but it's actually pushing more of it to the membrane as well. So we looked at, well, what happens to DRA when we have loss of CFTR function? Whoa, that's loss of PowerPoint function, or loss of CFTR expression in F508del homozygous tissue. And so what we see with CFTR inhibitors, you get more DRA at the membrane. With linaclotide, you actually don't get further DRA at the membrane, but within CF, you have more DRA at the membrane at baseline, actually, and then linaclotide stimulates even more DRA. We wondered, well, why don't we see more DRA at the membrane here? And when we looked at these are all normalized data, but when we looked at the actual mean fluorescence intensities, it seems like we may be already at sort of the peak threshold there. So we may not be able to put more DRA in the membrane because we already have achieved a lot here with just CFTR inhibition. So we've been able to show you that linaclotide increases duodenal bicarbonate secretion. It does so independently of CFTR. It also increases DRA at the membrane to accomplish this, but we wondered, well, how does linaclotide get DRA to the membrane? Typically, when we think about linaclotide or STA's activities, we think, like I said, it utilizes this guanylyl cyclase C receptor that increases cyclic GMP and can alter cyclic AMP levels and prostaglandins to do that. But the main effects are thought to be on CFTR and in increasing CFTR function and decreasing NHE3 function. And now we've added DRA to that list. Well, when we did our initial studies in CFTR knockout mice, we actually also had done studies giving them a cell permeable analog of cyclic GMP, 8-bromocyclic GMP, which normally stimulates very robust bicarbonate secretion. But in CFTR knockout mice, you don't see an increase in bicarbonate secretion. So we felt that in CFTR knockout mice, maybe you're utilizing a different intracellular signaling pathway to accomplish the DRA effects. When we go back and look at the literature, it's actually been shown that STA can increase IP3 and intracellular calcium. This is IP3 in the back here increasing within 90 seconds. And when you measure intracellular calcium and you give STA here, you can see an increase in the intracellular calcium pools, despite everyone thinking about cyclic GMP as the main mechanism. In our lab, we've also been able to show that when you give linaclotide in duodenal enteroids, you can see an increase in intracellular calcium, and this is with carbachol, sort of a similar magnitude as carbachol, although their dynamics are differently. And then in vivo, again, when we do these experiments where we have CFTR inhibitor present and linaclotide to look at CFTR independent bicarbonate secretion, when we give an inhibitor of PLC here, we can totally abolish that response. So we started to think about, okay, calcium may be involved, phospholipase C may be involved, and that got us thinking about this protein, IRBIT. So IRBIT is a shorter name for a much longer name. I'm not going to say that all the time here, and Julie's probably happy that I didn't make her say that. And this is the gene name ACHYL. Well, IRBIT really is a hub protein. It's involved in many different things, has many interacting partners. But what I'm going to talk about today is really about its involvement and its potential to move ion channels to the membrane. And it's thought to do that in one way is that you can activate phospholipase C, generate IP3, and normally IRBIT is bound to the IP3 receptor. But as soon as you get more IP3 and it binds to the IP3 receptor, it displaces IRBIT, and then IRBIT now shuttles things to the membrane, okay? And this has been shown to occur in the pancreatic duct, but in the duodenum, there was no data on this. So our hypothesis was essentially that in CF tissues, when you have linaclotide or STA, that this is what's happening. You're increasing IP3 generation, displacing IRBIT, and that's trafficking DRA to the membrane. So first we looked at IRBIT expression and localization in human duodenum. And what we find is that in duodenal biopsies, you have quite a bit of IRBIT expression, even compared to other cell lines like Calu3 or HEC cells. And here's a duodenal biopsy with red being IRBIT. You can see pretty robust expression. It seems a little bit more in the villus region than in the crypts here. When you look at single cell RNA sequencing data, we see a lot of cells expressed IRBIT, especially in the enterocytes. And interesting to us, when we looked at SLC26A3 or DRA, it seems to have maybe a similar expression pattern. And in fact, when we look at villus cells, specifically from single cell RNA sequencing data, and look at co-expression of DRA and IRBIT mRNA, we see about 40% of cells co-express both. When we look at duodenal enteroids, we can see, looking at RNA, and this time it's an undifferentiated crypt-like enteroids all the way to more fully differentiated villus-like enteroids. We can see that DRA, we know, increases in expression as you come up the villi. IRBIT also seems to increase it more in this villus region. We see that in basal out enteroids and also apical out enteroids as well. And when we look at a protein level via Western blot, we also see more IRBIT in differentiated enteroids, which sort of seems to fit with the biopsy data that we have. So going back to similar studies that I showed you with DRA trafficking, we looked at IRBIT expression. Now IRBIT is in red, and under controlled non-stimulated conditions, we see IRBIT throughout the cells. These are many cells within a single enteroid. And then when we give linaclotide, we can see more IRBIT is now at the apical pole. And when you zoom in some of these regions, the yellow is where IRBIT and vilin, which is marking the apical surface, are co-localizing, meaning there's more IRBIT there actually in the membrane. And when we quantify, we can see more IRBIT at the membrane when we give linaclotide. So what we've seen is that linaclotide can increase DRA at the membrane, and linaclotide can increase IRBIT at the membrane, but do they do it together, or is it just purely an association? So we've begun to do some of these experiments to look at this more, and this is preliminary data. So we started with co-immunoprecipitation, and when you IP with DRA and then look at immunoblot with IRBIT, you can see it. And likewise, when you immunoprecipitate with IRBIT and immunoblot with DRA, you also can see it, meaning that within the cell at a protein level, they seem to be interacting to some degree. When we look at immunofluorescence, this is now a duodenal biopsy, and whole mount mode is just a section through it. And again, sort of green here is DRA, red is IRBIT, and the yellow is where they're co-localizing. We do see areas that they seem to be co-localizing, and predominantly they're at the membrane. So we then went to do some of these experiments where we gave a PLC inhibitor, the same one I showed you before that inhibited the bicarbonate secretion. And when we look at DRA trafficking to the membrane, these are all now with linaclotide stimulation here. Without a PLC inhibitor, you get a lot of DRA at the membrane. You give the PLC inhibitor, you no longer get DRA at the membrane, okay? Interestingly, I'd also shown you that CFTR inhibitor alone increases DRA at the membrane. And when you give PLC inhibitor, that also decreases that. So it does appear that PLC seems to have a role in moving DRA to the membrane. Now, when we look at IRBIT, we did not expect to see this, but so far we see that there's no difference in whether you have PLC active or not. IRBIT still remains actually there at the membrane. And we haven't done experiments yet in CF on them. So just quickly here, to get more information on this, we're using CRISPR-Cas9 to generate IRBIT knockout enteroids. And so our preliminary data shows that we can achieve very high indels, 100%, and disruption here with using this multi-guide targeting of exon 3. So we're characterizing these right now to see if we actually get knockdown of protein. And so what hopefully I've convinced you of is that we have shown that STA and linaclotide can stimulate CFTR independent bicarbonate secretion through DRA trafficking and activation. And this appears to be PLC dependent. Linaclotide also increases IRBIT membrane expression. However, this may not be PLC dependent. And we're looking into this other mechanism whereby IRBIT through interactions with CAM kinase 2 may also be able to translocate proteins. So we have a number of next steps that we're looking at. We're going to look at IP3 generation, look at more into what's going on with IRBIT trafficking and CF enteroids. Also, like I said, look into the involvement of CAM kinase 2 in this pathway. And we're very excited to look at to see what happens in our IRBIT knockout enteroids, because there is no actual pharmacological inhibitor of IRBIT itself. And then we're also interested in how IRBIT may also be affecting other acid-based transporters like CFTR, NHE3, and PAT1. Thank you.